What I'd like to talk with you about today is the overarching regulation of what we have always referred to as genetically engineered animals, although those of you who may have attended some of the NAS meetings earlier this week and last week will know that there's some discussion now about what we call things. But let us, for the sake of argument, just refer to them as genetically engineered animals. I'm not going to spend too much time talking about the Federal Food, Drug, and Cosmetic Act because my colleague, Dr. Wildman, gave a wonderful presentation on that yesterday, and that'll be posted on the NAS website, I think probably within about a week. So I'll quickly reprise for those of you who were not there the FFDCA issues and spend a little bit more time talking about the NEPA issues and how we approach them from a risk perspective. This is going to be a more science-y type talk than it is going to be a legal talk because I am trained as a scientist, so that's where we are. Okay, so the statutory authority that the agency has for regulating genetically engineered animals comes from the Federal Food, Drug, and Cosmetic Act. And as Sarah said earlier, we regulate products. In other words, the law says we are regulating an article and not a process, although the process by which that article is made is something that is evaluated. And the law requires us to determine that the sponsor has indeed demonstrated the safety of the article and its effectiveness. NEPA, as Sarah mentioned, is a procedural act which orders agencies to evaluate the impacts of what are known as major agency actions. And in this case, it would be an approval, although we also have an opportunity for doing an environmental assessment at the investigational or research stage. Okay. One of the documents that we issued in 2009 and that went out for public comment in 2008 is Guidance for Industry 187, which you can find on our website, which clarifies our regulatory authority under the Act, and then the second part of it talks about how the regulations are how the recommendations in Guidance 187 are congruent with the regulations in 21 CFR 511. And then provide some recommendations to sponsors who are the people who own applications or developing the animal, can generate data that can help address safety and effectiveness. And although it's present at a very high level, what we really would like people to do is come in and talk to us as soon as they can so we can help them design the appropriate studies so that all of our risk questions can be answered. Some of the other take-home messages here are that this is a mandatory pre-market approval, and we have post-market oversight once an approval has been issued, and it covers all of the genetically engineered animals that are made regardless of their intended use. So it could be an animal intended for food use, an animal intended to produce a pharmaceutical in its meat, milk, or eggs, or an animal that is intended to be used for xenotransplantation or the production of other high-value products. And that we use a risk-based approach, which means we don't have a checklist of things that you have to do, but rather for each article, we ask a certain set of risk questions. So what is an article? And for us, an article in this context is a recombinant DNA construct intended to affect the structure or function of the animal. And that is, that is because it meets the definition of a new animal drug. It meets the definition of an animal drug, I'm sorry. And so what does that mean? Does that mean that the Aqua Advantage fish is a drug? No, it doesn't. It means that the Aqua Advantage salmon contains a construct that is regulated under the new animal drug provisions of the Act, which gives us a great deal of flexibility and a great deal of rigor in our review. It covers all of the animals that are descended from the lineage progenitor, and we define, if you look at Guidance 187, we define founder and lineage progenitor differently. You may need to do some traditional backcrossing to get rid of extra insertions if you're using classical microinjection or electroporation types of techniques to get constructs into the genome of the cell, although I think probably with the site-directed enzymes these days, we're going to have a little bit more congruence between the founder animal and the lineage progenitor. We do event-based, which means each insertion site is its own event and is its own new animal drug, because the animal drug is actually the rDNA construct at a specific site in the animal. And we do case-by-case evaluations because depending on the nature of the construct and its intended effect, the risk questions we ask may be very different. We have what's known as an enforcement discretion path, which means based on risk, meaning that we anticipate very little risk, that we will not ask people to go through the entire new animal drug application process, but instead answer the kinds of questions that will address our concerns regarding the potential risk from the introduction of that organism. And an example of that that's been longstanding is the glowfish. And then we have approval paths, which are actually do grant a specific approval for a specific set of use conditions that are specified in the application. Okay, so what is our regulatory trigger? I said at the beginning of this talk that we're calling different things, different constructs, different modifications or alterations of the genome, different things. And what I'm going to be talking about for this talk is how things stand today and not necessarily how they will be in the future with the advent of all of these emerging technologies. So the important thing for us is that the name GE or GM or genetically altered is not our regulatory trigger. Our regulatory trigger is, come on, slide, is the term drug. And there are 4 definitions of the term drug in the Federal Food, Drug, and Cosmetic Act, of which 2 are important to us. One is articles intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals. And that you can think of as what you usually get from the pharmacy when you don't feel good and the physician prescribes something for you. And in the case of genetic engineering, those could be gene therapies, for example, that are used in animals, or non-heritable constructs that are used to help treat a disease in an animal. The definition C, articles other than food intended to affect the structure or any function of the body of man or other animals, is the one that generally prevails for us, because the point of doing the genetic alteration is to affect the structure or any function of the body of the animal. So that's the article definition that we use most often. The question then comes, what do we mean by risk-based analysis? And for us, it's very, very important to distinguish between hazard and risk. Lots of things can be hazards, but in the absence of an exposure, they do not pose a risk. Where risk is the conditional probability of an adverse outcome provided that exposure has occurred. And so we're very careful to say, well, here's the characterization of all the hazards that this particular substance or activity has, but the risk estimation is very different because it's dependent on the conditions of use, which is a function of exposure. So what we do is we break the overall determination of risk down into individual steps. We try to ask the appropriate risk question for each step. We evaluate all of the available data that there are to look at, and then we do a weight of evidence determinations in drawing our conclusions that is based on data and information, and then we characterize the uncertainties that are associated with that. So this process, as Dr. Wildman showed folks the day before yesterday, and for those of you who haven't seen it, takes the form of essentially a ziggurat or a wedding cake or whatever you'd like to call it. Starts out with the product definition, in which we try to say we're looking at this kind of a construct and this kind of an animal that is intended to do that. And what it is intended to do goes back to the claim validation, because that's the effectiveness part of it. We ask for a molecular characterization of the construct prior to its introduction into the animal, a characterization of the construct in the animal and how it behaves in the animal lineage. We ask for a phenotypic characterization of the GE animal for two reasons. One is to determine whether or not the introduction of the article is safe to the target animal itself, and secondly, for purposes of environmental assessment, to see if we can make any kind of determination about net fitness of the population of those animals based on what we know about the phenotype. This part here, the genotypic and phenotypic durability plan, essentially asks the sponsor to demonstrate that the animals that they are looking at in the pre-approval stage are all behaving consistently with respect to genotype, that that's stable, and that the phenotype is stable as well, and then asks for the sponsor to develop a plan that's kind of a quality control plan to ensure that the animals that continue for the lifetime of the product life cycle are equivalent to those animals that were evaluated for the approval. Then we do the actual safety determinations. We do a and feed safety determination for those animals that are intended to be used for food. For those animals that are not intended to be used for food, we look very carefully at identification, method of identity, and containment, including appropriate disposition of animal carcasses and waste to ensure that none of these animals or their edible products enter the food supply. For the environmental assessment, I'm going to ask you to hold on for just a second. I'll get to that. And as I said previously, the claim validation is a measure of the effectiveness. What the sponsor wants to choose as an appropriate endpoint and one that we agree with or concur with is the effectiveness claim. So what is a weight of evidence determination? It considers all data and information. Unlike evaluations of conventional new animal drugs, we don't have what we call pivotal studies. We have studies that are very important, but we don't rely on a particular study unless it is required to do so in the act itself. This system uses a qualitative priority that's given to different sources of data and information, to which we refer to as deference. It's based primarily on the Hill criteria for causality that were published in 1954 and were used for human epidemiologic causality determination, but are quite amenable to any kind of causal or inferential pathway. And it allows us to have clear statements of uncertainty where we have them. So I don't expect you to read this table very carefully, but basically what this does is provide examples of what kinds of studies we give the most deference to. I'm sorry for that dangling participle. So the highest degree of deference goes to controlled studies that have the final structure of the DNA construct. They use the same animal lineage that we want to evaluate and have a high degree of internal validity. Those studies could be modified with respect to the amount of deference we give them based on the quality of the study, how many generations of animals you've looked at, and the relevance of the particular risk question that you're looking at. Obviously, you don't particularly care that much about morphological changes if you're asking a food safety question, okay? And the other important part here is all of these deferential criteria rest on a foundation of biological plausibility. If there's not a good biological basis for that, that means that we have to go in and look for what the possible causation is and make a determination as to whether or not it is important to our assessment or if it's just a numerical problem or a validation issue. So as I said before, each GE animal may pose it's a set of unique risks, so we have specific risk questions that we ask for each thing, and I imagine that if somebody brought a gene drive to us, we would have a very different set of risk questions than we would if somebody brought us a standard construct that is going to be propagated in the Mendelian fashion. Biased inheritance is very different from Mendelian inheritance. And again, as I said, we do a case-by-case evaluation, and the application itself drives the assessment that we do. So one of the things that's really, really important to understand is that when we approve an article under the new animal drug provisions of the Act, we do so for a specific set of conditions. In the case of the Aqua Advantage salmon, the particular set of conditions that was proposed in the application was for breeding at the Prince Edward Island facility, shipping fertilized triploid eggs down to Panama, grow out in the Panama facility, harvesting the animals in Panama, and importation of food from those animals into the United States. The forms of physical containment were highly specified. The forms of biological containment were highly specified, and all of those were validated by both rigorous data review and inspections of both the PEI facility and a site visit to the Panama facility. So we're ensuring that it's these conditions and these conditions only. I'm going to stop the FFDCA discussion here, and if you have any more questions, please see Dr. Wildman's presentation. So NEPA was passed in 1969. Its goal was to restore and maintain environmental quality in the United States. It specifies that federal agencies must consider the environmental impacts of their major actions. Those actions include drug approvals, such as the approvals of genetically engineered animals. And the question that's asked by the Act is, will the action have a significant effect on the human environment of the United States? Actually, it's a significant impact. So here are the NEPA regs that were established. Most of them come from the Council on Environmental Quality, but FDA has its own set of regulations on environmental impacts, and those regulations address specifically the preparation of environmental documents and address the issue of public participation and notification. So effects, as I said, are actually impacts. They can occur in two types: direct effects, which occur in about the same time and the same place as the agency action, and indirect effects that may occur later in time or removed in distance, but must be reasonably foreseeable. And it's later in time or removed in distance from the actual the agency action, okay? So this considers ecological effects and potentially others as relevant for the action being considered. So how does one proceed in environmental assessment in meeting NEPA requirements? And what I really want to do is find the slide that I cannot find. Okay, so the environmental assessment is based on evidence and analysis to make a determination on the potential for significant impacts and whether to, after that assessment, to prepare a finding of no significant impact, or if we do determine that there is a significant impact, to move on to an environmental impact statement. It allows the public to understand the agency's decision, and it's supposed to be a concise, objective, and balanced document, although I think one can argue that the 169-page environmental assessment that we issued for the Aqua Advantage salmon is not what necessarily, well, concise would be in the eye of the beholder at that point. So what are we responsible for when generating an EA? The agency is ultimately responsible for the scope and the content of each EA. Normally what happens is the sponsor prepares a draft EA that we take a look at, and then we determine whether or not that draft EA is sufficient to serve as, to stand in for the agency's analysis, or if we wish to do our own environmental assessment, which is what we did with the Aqua Advantage salmon, although the sponsor did present their EA, and it was discussed at the Veterinary Medicine Advisory Committee meeting in 2010. Following the approval of the regulated article, if we get to the point where we determine that there are no significant effects and a finding of no significant impact is generated, both of those documents are made available to the public through the docket. Now, again, the Aqua Advantage salmon environmental assessment and the FONSI were an exception in that the public comment was requested on both the draft EA and the preliminary FONSI. We had a 120-day public comment period in which we did ask for comments before we finalized the EA and FONSI and posted them at the same time that we posted the approval. So in summary, an EA is required when a new animal drug approval is to be granted. The environmental assessment is the basis on which a determination is made, and if a proposed action is without precedent, the EA and FONSI will be made available for public comment before the agency makes its decision. The important take-home message here is that the environmental assessment always considers the conditions that are specified in the application to be approved. It does not do a programmatic EIS. It doesn't look at all potential uses. It looks at the particular uses that are to be approved in the application that is to be approved. So we have a conceptual model for hazard characterization and risk assessment. I'm not going to go through this except to point out a couple of things. One is that you have to make the determination as to whether or not you are going to have contained use or you're going to have free release. The accessible environments need to be evaluated. Then we determine the ability to survive, disperse, and establish, which are all kinds of functions of net fitness. And then we look at the direct and indirect effects and the impacts on populations, communities, and ecosystems. We can spend a lot of time going through that, but I think it's clear from this that this would be a case-by-case approach, and this outlines the steps that we would take in doing an environmental assessment. Okay, so the information that we evaluate comes from the product definition and the conditions of use in the application itself, including containment if it's required. We take a look at the sponsor's draft EA if they have provided one. We inspect the sponsor's facilities. We validate containment if it's important or required. And then we make sure that we've characterized the phenotype with respect to fitness, as I mentioned previously in the FFDCA analysis that we do. And we use the weight of evidence determination when we do the environmental assessment as well. So with that, I'd like to thank you for your attention. I hope I didn't go too much over time. If you have any questions, you may contact me by email, and I think Dr. Pearson has the floor now.